Okay, we got the last little section to talk about. We're just going to really hit the high points here. A lot of this is about DNA manipulation. We'll just give you, again, the highlights, and a lot of this will come up later. So let's jump into it. And since we've talked about DNA so much, and we're starting to get this picture that it's important for genetic information, the question arises, well, do we know the sequence, or how can we figure out the sequence? And the answer is we do know the sequence, and we will talk about some ways that you can actually sequence DNA. And why this is useful is that from the DNA sequence, we can figure out what the mRNA and eventually what the protein sequence is. We can start to look at whole genomes, and we can predict which parts of the DNA, which genes are important and what they do, and start to identify areas that may be causing disease or be related to disease. We can use it in forensics chemistry to identify criminals. And also we can use it to identify unknown organisms. So you can take a scoop of soil and sequence and start to figure out what type of organisms are around us that maybe we can't culture. So there's actually lots of different things that you can do. And I've got a little picture down here actually showing a sequence of DNA. And so we're going to talk about how you can get this. You'll notice that there's colors and peaks, and at the top there are letters. And so this is the actual sequence that we're reading up here. But how do we get these peaks? And so we're going to talk about that after we talk about the first sequencing and why we need better ways to sequence than just structural determination. And what I mean by that is if you took a strand of DNA and tried to figure out what a sequence was, well, first thing would be to do is the saying, like, how do you eat an elephant? One bite at a time. So how do you sequence DNA? Well, if you could get it down to one base at a time, that would be nice. So if we've got a big long sequence, you want to break it into smaller pieces. And so one of the first ones, Robert Hawley, took him 7 years to sequence a tRNA. And what they did was they took a snake venom phosphodiesterase. This is an enzyme that cleaves nucleotides, and it cleaves somewhat randomly. And so they, well, sorry, not randomly, but it'll chop off, nucleotides kind of one at a time. And so they let it run for a certain amount of time. So they end up with a bunch of sequences there. And out of this, they would separate these sequences. And we've talked a little bit about how you can actually separate the sequences. And you imagine if I just get it down to a dinucleotide, I might be able to determine what that structure is chemically or through some other laboratory methods. And then I find the piece that's a little bit bigger. And if I can just find that one piece of information, I'm one step closer. If I've got this other small piece here, then I can solve that problem. But it still was very tedious because they had to take many, many different pieces and figure out what their structure were. And then eventually they could assemble it back into the sequence. So they actually discovered restriction endonucleases. These are enzymes that cut in the middle of DNA, and these were discovered around 1975, and they can actually cleave at very specific and well-defined spots, and we're going to talk about that a little bit more. But we've seen this picture before, and so A through H is actually the same strand of DNA, but it's been cut by different endonucleases. And so it cuts into characteristic patterns. In fact, this over here is a standard. So they know the sequence of that DNA and they cut it with a specific endonuclease, and you end up with these markers here. So now I can take this marker here and figure out the approximate weight or number of bases in that sequence there. Anyway, this was just a lot better way because you could actually start to get fragments of defined size here. And so again, all these here, it's the same sequence, it's just been cut by different endonucleases. And that will be important because this sequence here is actually going to have some similarity to some other sequence over here, and we'll talk about that. So if I start sequencing these, then what I need to do is assemble the entire sequence, and so I have to overlap them. But if I can take little small bites and sequence them, then eventually I get enough overlap that I can reassemble the entire sequence. Okay, so a little bit more about restriction enzymes. They call them restrictions because bacteria actually have somewhat of an immune system. They can actually have methods to try to prevent the attack from foreign DNA. So viruses like to inject their DNA into bacteria. Bacteria don't like that so much, and so bacteria have developed these restriction enzymes that will cut up specific types of DNA from bacteria. They also do some other interesting things to get kind of a memory of being attacked by viruses. It's a lot different than the human immune system, but it's led to some incredible technology such as CRISPR-Cas9 technology. We may get to that a little bit later. But these restriction enzymes, there's different types, but type 2 and type 3, they cleave DNA at selected sites, so they're very specific. And what they do is they recognize either 4 or 6 bases. So they scan along, they'll find 4 or 6 bases, or even more that they recognize, and then they will actually cut there. And just as a side note, a restriction enzyme that recognizes 6 base would be known as a 6 cutter. So type 2 restriction enzymes, they don't require ATP. The recognition site in double-stranded DNA have a twofold axis of symmetry. And I'll give you a more detailed picture of that in just a bit. Cleavage can lead to what they refer to as sticky ends or blunt ends. I'll give you a picture of that in just a second. And they just name them using a 3-letter italicized code. And so it just comes from the genus and the species of where they found these enzymes. And there may be some other letters and things that denote the strain. So for example, EcoR1 is the first restriction enzyme isolated from the R strain. So that's where the R and the 1 come from of E. coli. And so the E comes from the E and the coli from the CO from the coli. There we go. Okay, so when we look at this, when we talk about that they have a twofold axis of symmetry, we need to introduce you to a palindrome. And so a palindrome, my favorite one is a man, a plan, a canal, Panama. So a palindrome is something that can be read forward and backwards and means the same thing. So if you actually read that saying backwards and you put spaces at the appropriate places, oh gosh, I can't draw. Man, plan, canal, Panama. So if we read it backwards, it says the same thing as going forwards. So in DNA, these palindromic sequences would read the same going from the 5' end to the 3' as it would from the 3' to the 5'. And then what they do is they can either cut what I'll show you this one off to the right here, blunt, so they just cut straight across. So you get a double strand break and you just get what are referred to as blunt ends, or they end up cutting between a specific nucleotide, like between the G and the A here, and also down here. And then what happens is you end up with these as sticky ends. So imagine if you took those blue and green parts away, they actually have some overlap and they could rehybridize, and that will be important for some DNA technology later on. So it depends on the type of restriction enzyme, but there's a whole list of them. You don't need to memorize them, but those restriction enzymes will recognize specific sequences. So here's a list of some of them, and this is showing where they cut. And then, of course, we can use gel electrophoresis that we talked about before to separate out these bands. And again, you can imagine that each one of these here is a different restriction enzyme. And so we'll get different size pieces. And that different size pieces will be important because large strands of DNA will be somewhat difficult to sequence. But if we can get small pieces, that will be much easier to sequence. So let's talk about one of the earliest ways to do that. But we need to talk briefly about an enzyme because this enzyme is important for making a complementary strand of DNA, and it's referred to as DNA polymerase I. And so what it'll do is it will take a nucleotide triphosphate here and add it on to the 3' end. So that oxygen would come and attack the phosphate, kicking out 2 phosphates and make a new bond. And so this occurs with an enzyme called DNA polymerase. And so the DNA would come along and then join the next one, and notice that this is complementary there. The process is a little bit more complicated than what we're seeing here, and that will be discussed later. One thing to notice is you do give off this pyrophosphate right there. That is useful in a type of sequencing. So Sanger came up with this method to sequence, and what he did is he used a dideoxynucleoside. Notice there's no 3' hydroxy here. And so imagine you take and you want to replicate a strand of DNA. And in that mixture, you have small amounts of these dideoxy nucleotides. So in there, you've got these nucleotides that don't have a 3' hydroxy group that can grow. So you have the regular, and that's what this is. You've got the regular deoxynucleotides, and then you've got a small amount of these dideoxynucleotides in there. And so you put it in the mix, and then you let the enzyme do its job. And then what happens is you get random incorporation of these dideoxynucleotide triphosphates. And once you include that in the strand, it stops. Now, what's important to think about here is that you've got millions and millions and billions of strands in there. So when you do this reaction, you're going to get all possible terminated sequences. Now, off to the right here, what we see is an experiment, and you've got 4 columns. Each one of these columns, it's the same DNA that they're trying to replicate, but in each column they added, or each experiment they added a different dideoxy. So here they added a dideoxyadenosine. And so what you see is different lengths along here. And this corresponds to when the chain stopped growing. So this is different than a restriction digest. What you did is you synthesized the DNA and it stopped growing at certain lengths. And you do that for all the nucleotides, and you can actually start to sequence it. And so what you do is you know that this one here stopped with a T. This one here stopped with a G. It looks like this one here stopped with a T. And notice this band's a little bit darker, and that's kind of an indication that maybe there were 2 T's or maybe even 3 different T's there. So far our sequence would be T, G, probably TT, and then it looks like AA, and then maybe a C, and then probably back to an AA, and this one right here is probably where it terminated with the first A, and then this one with the second A. So it's not perfect. It doesn't give you a good read, because if you have examples where you got two A's together, it's going to be a little bit harder to see there. So they use this. This is what's referred to as Sanger chain termination DNA sequencing. Now, what they ended up doing is, instead of using that previous method, they actually put a fluorescent dye on these dideoxy. Notice none of these have hydroxy groups down here. And so you've got different dyes, different colors on there. And so they use this in the same process. They'll mix it in. You've got your regular DNA triphosphates, but you have a small amount of this dye. And the same process is going to happen, and that is you're going to terminate these sequences at random positions. And then what you do is, in this example here, is like what we saw before, where you run the gel and you can figure out the sequence. So I could figure out that this is A, T, A, C. But there is, like I said, you'd have to kind of manually read that off. But if you could run this down a column and separate them based on size, and then have a laser shine in and give you a color, you can automate this. Now, I know that this picture and this picture matches up. This one off here doesn't match up with this one here. So let's just look at these two here. So you can automate it, and what you do is, the smallest one will run out first, and you got a laser, and as it comes out, it gives you a green color. Then as it all passes out, the next one comes through and it gives you a red color. So essentially you're just running this down a long column, and the products are coming out the other end, and a laser is exciting it. And every time you see an increase in that fluorescence, and you know the color, you know what type of nucleotide that is. And so what we can do is start reading and assembling the sequence from this pattern of colors there. So here is, off to the left, is a little bit more informative picture. So what you do is you put a primer. You've got to have a starting place for the polymerase to work. And then what you do is, in some cases, it will add a regular A and a regular A and a regular G and a regular A, and it keeps going and add a regular G. And then what it'll do is it will add the dideoxy C, and then it stops. And it would give you that molecule. And now that molecule is sitting in solution. It can't grow anymore because there is no 3' hydroxy. In another sequence, it grows and it adds all the nucleotides. And at some point, it adds the dideoxy A. And actually, sorry, I gave you the wrong one. If it adds, it's not adding a dideoxy A, it's adding a dideoxy T. It's got to add the complementary one. So it would give you this strand right here. So my previous one, I should have cut onto that, but anyway. So it's adding, and eventually here, it's trying to make the complement. So it would add a dideoxy T that's complement to the A, and that gives you this strand right here. And that's just floating around in solution. So let me correct my error one more time. So you're adding just the complementary nucleotides. So here you're adding a T and a T and a G and a T and a C and a G. And then all of a sudden you add a dideoxy A. And the chain can't grow anymore. And so this is made in solution. So randomly, you're going to get termination at every single one of these steps. And so in solution, again, we're dealing with millions and millions of molecules. You have a representative of every terminated sequence there. And only the terminated sequences have the dye on there. And so as you run it out the gel, the smaller ones come out first. And so you would see the T, and then you'd see a T. Then this next sequence would come out, G. And again, what you see actually experimentally is just this absorbance over here, but the computer will assemble that into a sequence for you. And so you can see that picture that we started with at the top. And so you get these colors and it will assemble that sequence for you. So that's Sanger sequencing. It actually can sequence for quite a ways, 700 to 1,000 base pairs before it just starts getting too messy and you really can't tell the difference. You can see that there is some noise in here. You're starting to see a little bit of different nucleotides, but since this is noise and that's a signal, you can be pretty confident that that's a T. But as you go along, you know, you look at this example here, and yeah, we're starting to get several different ones coming out right there. So maybe you just have some DNA that's a little bit shorter or it was misincorporated. But the longer you go, the more likely that is to occur. And so eventually you'll just get kind of just a mess of these coming across at the same time, and you can't figure out which one's which. But again, 700 base pairs to 1,000, that's pretty good read rate. But there are next gens that can do this faster. And so they end up doing this. They tend to use smaller fragments, but they do thousands to hundreds of millions of reads. And so this is what's referred to as massively parallel sequencing. So they have shorter read lengths, but they're reading many more sequences. And so here's just an example of an Illumina next-gen sequencing. So it uses 2 sets of nucleotides. It uses one that can react, but caps it and stops it so you don't get multiple growth on there. And then it uses one that has a fluorescent tag that can also be removed. And so what you do is you go in and essentially run this process. And so they're using fluorescent tags. But if you add in these nucleotides, what you'll see is colors appear at different spots. So right here is one sequence of DNA. And in cycle 1, you can see that we've added a C. This is a different sequence of DNA, and you can see that we've added a G. Then what they do is they remove the dye. And so actually, if they had the intermediate pattern here, it would just be all black because there's no more dyes on there. And then they add the next nucleotide. Well, you can see if we follow this one, it goes from green to blue. So its sequence would be C to A. And then it goes C-A-T, back to C, back to, or over to G, and then back to T. And so you just have a good enough camera that can focus on all these spots and watch the colors appear, and then that will give rise to the sequence. Now, I posted some videos in the other information, and there's a great little short video. It's only a couple minutes on this Illumina next-gen sequencing. So I'd encourage you to watch it, and it'll explain it a lot better of how they sequence. So there's other ones in there. There's the Oxford Nanopore, and just as an interesting side note, I did my postdoc in the group that developed that. I was actually in the lab when they started doing some of those first experiments. It's too bad I didn't stick with it and stay with them. I could be a millionaire now, but I'd rather be with you. So they can sequence entire genomes using these methods. And they've done it for humans. And you can see that that is 3 billion base pairs. That's a lot of information there. And it took them a long time to do it. So in fact, the group that I was working for in England, they had received a huge grant, and the goal was to get to sequence a human genome for $1,000. It was called the $1,000 Genome Project. And they can pretty much do that now. If you wanted to sequence your entire genome and you had $1,000 lying around, you could likely get close to getting your entire genome sequenced. And some of the interesting things that you can find out when you start sequencing DNA is the relationship between different organisms. And so Oh, I looked up how to pronounce this. I'm not very good with Spanish, but Tietz- Tioxetine. Oh, I just murdered that. Maize. I can say maize. And this other one. These only differ by very few genes. And so it was thought that early Americans, specifically around Mexico, they bred and converted or selected for genes that caused this little kind of wheat grass to actually turn into what we know as corn on the cob. And so they only differ in a few genes. And so you can actually see where they differ. Okay, we can also use sequencing to actually find places in our DNA that are associated with certain diseases. And here's just a few of those. Okay, so we can sequence DNA. And like I said, there's some very good videos that talk about that, and they're really short. So I would take a look at those. And we can manipulate DNA. I'm going to go through this really quick and tell you the high points. And you can read this as well as I can, but why would you want to clone a gene? So cloning is actually isolating, amplifying, and modifying, or essentially manipulating the DNA sequencing. So you want a bunch of this DNA to study or to put into some other organism. And so here's 6 reasons to clone a gene. But the 7th, I think, is the best, that you want to make a fluorescent cat. So this type of sequencing and cloning allows us to do interesting things like introduce the green fluorescent protein that you found from, oh my goodness, someone email me the answer to that. I can see the organism. I just can't think of the word. They live in the sea and they have the green fluorescent protein. I want to say squid. I know it's not squid. Anyway, someone emailed me just say the one word, the organism that they got green fluorescent protein. I'll give you 1 bonus point. Okay, that'll let me know that you're listening as well. But you can include it in cats. This cat isn't glowing green. It's just at a rave and it fluoresces green. So that is pretty cool that we can do that. So you can manipulate DNA. So you can take a fragment of DNA. You can increase the amount by PCR that we'll talk about in a second. You can incorporate that DNA into a carrier that we refer to as a vector. You can put that vector in an organism of interest and change that organism or have that organism make more of that DNA for you. And then in order to do this, you actually need to be able to make sure that that cell has that DNA of interest. And so one of the ways to do this, and this is just one, is to use a plasmid. And a plasmid is a circular piece of DNA. These come from bacteria. And here is a plasmid that is commonly used. There's lots of them, but they include a couple important parts. They include what's referred to as the origin of replication. That's where we're going to start replicating this DNA and making new copies of it. You need a selectable marker, something to let you know that you have included your DNA of interest. And in this case, it's going to be this LAC-Z right there. You need multiple cloning sites. And what that means is you have a bunch of these different restriction sites. And so I could include different fragments of DNA into a plasmid. But the key is that these multiple cloning sites right here, they need to have different restriction sites. And these are other, in this plasmid here, they're just identifying other restriction sites. So I don't want to cut over here. I want to cut in this region here. And I want to cut in this region here because when I go to make this DNA, I'm going to make this sequence right here. And I said, this is the reporter region, and I'll show you what that does in just a second, but I'm going to include my DNA in that region right there. And you can include other stuff. So this polylinker, or this multiple cloning sites, is an area that has a bunch of unique restriction sites. So they don't occur elsewhere in the plasmid, just in that part. And so it allows you to selectively insert the DNA right here. And that's very important. I don't want it to be randomly incorporated elsewhere in the plasmid. I want it to go in a certain spot. And then what you need is a promoter, and that needs to be upstream, meaning that it needs to be ahead of where the new piece of DNA is going to be, so that when I go and replicate this DNA, I make sure to replicate that new piece of DNA that I inserted. And there's a couple different types of promoters, one that's always active, one that you can induce by changing conditions. And then you need a place that this is going to stop, so that it's going to not continuously replicate these plasmids. Okay, so let's talk about how you know that you've inserted the DNA. So in there is this LAC-Z gene, and it makes beta-galactosidase, an enzyme that cleaves this molecule right here. So if I don't insert DNA, it makes the enzyme that cleaves this, and when it cleaves it, it turns blue. So if I do nothing, I make colonies of bacteria that are blue. If I insert my new piece of DNA in here, it's no longer going to make the beta-galactosidase, and so the bacteria are just going to grow up and they have kind of a tan color. This is often referred to as a blue-white screen. Blue is bad, means that you didn't transform your plasmid or your organism. White means that you did, means that you inserted a piece of DNA here and it no longer makes the enzyme because I've inserted or I've cut that piece up. And again, this is going fast. It's just kind of giving you an idea of what you can do. So what we really need to take home from this is, yeah, we can manipulate DNA. And you can actually insert multiple genes and lots of different things with them. So in fact, plasmids can carry up to 20 kilobytes, or sorry, not kilobytes, sorry, kilobases of foreign DNA. So you can actually use it to add an entire biochemical pathway to a host organism. So it's really interesting. A lot of the drugs that we make in the laboratory, they try to get organisms to make them, because organisms are, they work for a lot less. Essentially give them some nutrients and they just chug along. They don't complain. They don't file workman's comp. They don't need to be quarantined, and they just don't complain. They don't need health insurance. So if you can get an organism, a bacteria or a yeast, to make a drug for you, it's much more cost effective. And so how do we actually get that DNA in there? Well, we've got this cloning vector, a plasmid, and we take advantage of these restriction sites in there. And like we said, those restriction sites are very specific, and they cut, if you get the right ones, with these sticky ends. And what you want to do is make sure that this sticky end here is complementary to that sticky end. And so if you use a restriction enzyme to cut the plasmid and cut your foreign DNA, and you use the same restriction site, then those sticky ends are going to match up. And then what you do is you just incubate them, and those sticky ends are going to find their way and start to base pair there and start to base pair there. And after a little while, you add in another enzyme called the ligase, and that just makes a covalent bond. We need these to be covalently bound so that you actually have that sugar phosphate backbone. Originally, when they just first base pair here, they're just going to be held together just by those hydrogen bonds right there. But in order for this to work, you need it to be a continuous sequence of DNA, and the ligase does that. So there's other ways to do this. You can use bacteriophages. These are actually viruses that inject E. coli or other bacteria with genetic material. And so what you can do is you can actually get these bacteria, you can load them up with DNA, and the DNA of your choice, and then they'll inject it into the bacteria, and you can make what are known as bacteria artificial chromosomes. That's what the BAC was referred to. So you make your own interesting piece of DNA, either by synthesis or some of these other techniques. And then you actually get it to package into the phage. We're going to avoid some of that. There's a lot of details here, but just understand there's lots of ways to manipulate DNA and get other organisms to take up that DNA and process that DNA. So we're talking about packing the DNA into bacteriophages, and so you can make a bacterial artificial chromosome. That's what a BAC is. You can also make yeast artificial chromosomes, and those can actually be fairly large. I'm not sure the number on them, but much larger than what you can pack into bacteriophage. So some of the advantages to these BACs, or bacterial artificial chromosomes, are they're similar to plasmids but derived from a fertility plasmid with bacteria, and they're more stable than plasmids. And anyway, a few other additions. Yeast artificial chromosomes, they need some different elements than a plasmid. So yeast are eukaryotes, and so they're not very stable unless they're large. So like I mentioned, the yeast artificial chromosomes, they can be very, very large. Okay, so here is a way to actually transfect and make a copy of a library of DNA. And so what you do is you grow up some DNA that you have hopefully inserted a desired sequence into. Now, you're not sure which of these colonies may have that sequence, or this may be a case where what you've done is you've inserted lots of different sequences into many different colonies, and you're going to want to find the colony that has a specific sequence. So there's lots of ways that you can do this. But in the end, what you do is you let the bacteria grow up on an agar plate, and then you have this handle with a piece of velvet on it. And what you do is you press it on the colony, and then you take that, what's been transferred to the velvet, and you put it on a nitrocellulose filter. And we've talked about this before when we talked about southern blotting. Is DNA, or single-stranded DNA, has very high affinity for nitrocellulose. So you put these complete bacteria on the nitrocellulose, and then you treat it with NaOH, and it does a couple things. It breaks down the cells and also denatures the DNA, so you get single-stranded, and then it binds very tightly to the filter, this nitrocellulose. And then what you can do is, kind of like you do in a southern blot, you wash over a labeled probe. And that label probe, you know the sequence of, and it has some type of label on there. Either it's radioactive or it has a fluorescent dye on it. And what it's going to do is find its complement. And you'll find its complement and base pair, and then you wash away everything else. And now what happens is, you know, this colony and this colony have the sequence. Then what you do is you go back to your master plate, and you have this index, so you know where those colonies are. And so those two colonies right there have that sequence of interest. And then what you do is what's called picking a colony. So you go in and you pull off a little bit of that bacteria there, and you put it on a new plate. So you take some from there, put it on a plate, take some from there and put it on a plate. And in reality, you'd probably take several different samples from each one of those and put it on many different plates, and then you let those grow up again. And then what you do is you do a little bit more checking and make sure that it has the complete sequence that you're interested in. So this is just a way that if you're trying to grow up lots of different types of DNA, you can do that. And so you can imagine that you could take this velvet plate and put it on a different nitrocellulose. So you're not going to transfer all the organisms. So essentially what you do is you'd stamp it across, 4, 5, 6, 7, 10 different nitrocellulose filters. And then you do the same process, but each one of these plates would get a different probe sequence. And so then let's say that this lit up, those colonies lit up there, these colonies lit up with that probe sequence, those colonies lit up with that probe sequence. In this example here, it's these that end up lighting up, and then those two colonies end up. So what's nice about this velvet is you can actually transfer a lot. You don't need much DNA for this to work, so you can transfer it to many different nitrocellulose, and then you do this hybridization experiment, and you could find out which colonies has different sequences of interest for you. And then, like I said, you go back to your master plate and you pick off those colonies, and then you regrow them up. Okay. Now, if you haven't had genetics, this may be a little bit fast for you, but we'll talk more about PCR, I believe, later. It's an incredibly powerful technique, and I'm a little saddened that they don't put more emphasis on it in biochemistry. It's kind of a molecular biology technique, so maybe they're assuming you pick it up in cell and molecular biology. But it's called the polymerase chain reaction. That chain reaction is important. It's a technique to amplify or copy specific segments of DNA. And so for PCR to be successful, you need a DNA sample, and that's going to be the one that you want to copy. And you don't need much of it. You could just need a few nanograms. In theory, actually, all you need is one sequence for this to work. And then you need DNA primers. These are going to be short sequences that hopefully have a pair, some matching point on the DNA sample that you're looking at. Then you need the raw nucleotides, and then you need a polymerase, one that's especially heat stable. And that's what's going to be important here. So DNA is naturally double-stranded, and so what you need to do first is actually heat it up, and that will separate the strands into their individual components. And then you slowly cool it, and you hope that the primers find the right spot. And so you have these big long strands that are separate, and then you have these primers. And so as you cool it, hopefully these primers find the right spot. And so that's step 2, that's the annealing, when the DNA primers bind. And once you get those to bind, then you raise the temperature a little bit just so you can get the DNA to do its job. And then the DNA will find the beginning of those primers and actually run along and make a copy. And then what you do is repeat. So lather, rinse, repeat. So now you go back to step 1 and you heat it up. And when you heat it up, that's going to separate those two strands. And then you go and anneal. In annealing, you will get those primers to find their spot. And then what you do is you run the polymerase again. Now, I only showed 2 examples, or sorry, 2 strands here. So this one actually gives 2. So if I did this whole thing right, and I'm not going to switch colors, but you'd actually go from 2 to 4. There we go, 4 double strands. And then in the next one, you'll take those 4 double strands and you'll separate them out into 8. So once you're done, you'll have 8 strands, and then you go to 16 and 32 and 64. And so you can see that rapidly over several different cycles here, you could increase dramatically the number of DNA you have in there. And so if I just had, like I said, literally you could just have 1 strand and amplify it to billions over several cycles. In fact, you could calculate how many going from 1 strand to get to 1 billion. This is kind of like that story about the guy that did a favor for the king, and the king says, well, I can either give you a bag of rice, or I'll put a piece of rice on 1 square of this chessboard, and then on the next square, I'll double it. So the next square has 2. And then the next square would have 4. And And so I'll ask you, which one would you take? Would you take the bag of rice, or would you take this where the king would take a piece of rice on a chessboard and then double it on each of the adjacent squares? And I can't remember the answer, but essentially, if you took it from the chessboard example, you would have enough rice to feed your family and the community for 1,000 lifetimes. I might be exaggerating there, but anyway, it's a huge amount. The better deal is to take this when you're actually exponentially increasing versus the bag of rice. And so that's what happens in the PCR, is you exponentially increase the number of DNA strands in there, so it grows. And you can look at this book or this example in your book. It essentially goes over what I talked about. I'll try to post actually a good short video. It's nice to see this in action, so I'll post that in the additional information for chapter 1 to 3 that will talk a little bit more about polymerase chain reaction. And that is pretty much it. There's a little section on how you can use DNA to identify criminals. I'm going to skip over that, so we won't be tested on it. And then the last is site-directed mutagenesis. How can I actually, let's just say I wanted to change one base in a sequence of DNA. And you can actually do that through PCR here, or there's other methods to do it. We're going to leave it off. I think that's enough information to worry about. So for this last lecture here, the important thing to understand is that we can amplify DNA through PCR. We can clone DNA, meaning we can move it from one organism to another and grow up multiple copies of that. And we can modify DNA. We can change its bases. We can put it in different organisms. And the specifics for that, like I said, are outside the scope of this chapter, and there's chapters later on. I believe it's more in biochemistry too, that you talk more about DNA technology and get into the very particulars here. So ultimately, the take-home message is DNA is very powerful because it's genetic information, and we have learned how to read and write and manipulate that genetic information for good and for bad. And if you think of a fluorescent kitten, I think we could all agree that that is for good.